Okay, so the review for exam 4, we'll start with chapter 18, which deals with amino acids. If you have a question, just feel free to unmute your mic and ask me, or you could type it into the chat, and I'll try to keep up with that. First question says, amino acids are the building blocks of proteins. Which of the following statements concerning amino acids is false? Let's take a look at all of them. A, it says that there are 20 different amino acids that occur naturally in human proteins. That is true. There are 20 natural amino acids. The amino acids that make up human proteins are called essential amino acids. That is false. Okay, so it's asking us for a statement which is false, and the answer is going to be B, because when it comes to essential amino acids are amino acids that we cannot synthesize. So I'll put here, essential amino acids are amino acids that humans cannot synthesize. So we're not able to make these amino acids on our own, synthesize, and so they must be taken in our diet. Let's take a look at the rest of them just for fun. It says the amino acids present in human proteins are alpha amino acids. That is true, except for glycine, where R is equal to H. Amino acids are chiral molecules. D is true because if you look at the structure of an amino acid, it goes NCC. We have our carboxylate group on this end. We have our ammonium group on this end. We always have a hydrogen here, or almost always have a hydrogen here, and then we have an R group. When that R group is a hydrogen, this is called a glycine molecule. So this is glycine. That is one of the amino acids. In fact, glycine is the simplest amino acid, and glycine is the only achiral amino acid. So this is not a chiral center. And you probably remember that a chiral center was a carbon that was bonded to 4 different groups. And since we have 2 hydrogens and they are identical, glycine is not chiral. So that means that D is also true. And then finally in E, it says amino acids are classified by the nature of their side chain R groups that are present. And that is true as well. And we'll look at that in some more detail as we go along. Number 2 says, or asks what term is used to describe the amino acid that has a free carboxylate group in a peptide. So if I have a peptide, for example, I went over this in my videos, how to draw a peptide. Let's say we make a tripeptide, you'd have NCC, NCC, NCC. So the tripeptide will have a carboxylate on one end. We'll have our ammonium on the other end. We have a carbonyl, so it goes NC carbonyl, NC carbonyl, like that. We're also going to have a hydrogen here. We'll have a hydrogen here on these carbons. And then we'll have our R groups. We have 3 R groups. So this is a tripeptide. I have 1 amino acid, 2 amino acids, 3 amino acids. And the amino acid that has the free carboxylate group, that we call that the C-terminal or the C-terminus amino acid. So this is the C-terminus, or you could call it the C-terminal. So right here, we'll call this the C-terminal amino acid. Okay, and hopefully you'll also notice that in my videos we say that the amino acid on the far left, this is the N-terminal amino acid, like that. Let's take a look at number 3. It says, what are the 2 most common types of secondary structure in proteins? And for the exam, you should be fully aware of primary, secondary, tertiary, and quaternary structure. Primary structure is just the result of peptide bonds or amide bonds. It's just the bonds that hold amino acids together. But once you get a number of amino acids together, they can have secondary, tertiary, and then if you have two or more amino acids that have tertiary structure, we can have quaternary structure. And the two common types of secondary structure are alpha helices, and beta pleated sheets. Beta pleated sheets. So you should be aware of those. And so the answer to this one is B. All right. And then in terms of tertiary structure is when we have folding of the peptide chain in on itself, and we have salt bridges and hydrophobic interactions, hydrogen bonding, and disulfide bridges. So all of those in here, we could even write those. We could put H bonding, so hydrogen bonding. What else? Disulfide bridges. There we go. We could put what else? We could put dipole interactions, dipoles, and we could also put salt bridges. So we could also put London forces or hydrophobic interactions, hydrophobic interactions. And those are the same kind of forces that will hold the quaternary structure or maintain quaternary structure when we have two or more peptides. Number 4, it says the amino acid 5-hydroxylysine is found in collagen. Which of the following represents this unusual amino acid? Well, I would give you a table of amino acids. I don't expect you to have the structures of all of the amino acids memorized, but I'll just help you out here a little bit, and we'll start by drawing the structure of lysine. Again, you don't have to memorize the structure of the amino acids, but in this case, it's kind of handy to have it. 1, 2, 3, 4. 1, 2, 3, 4. So losing count here. There we go. So this, what I've drawn over here, this amino acid is lysine. This is a basic amino acid, lysine. Okay? And the way that we number the carbons in the amino acid is that the carbon in the carboxyl group, that is always going to be carbon number 1. So we have 1, 2, 3, 4, 5, 6. So we have a total of 6 carbons in lysine, and this is how they are numbered. So if I look at all of the possible answers that are here, I'm looking for one that has a hydroxy on the 5th carbon. Now a hydroxyl or a hydroxy is an alcohol. So which one of these has an alcohol on the 5th carbon? The answer is number 3, right? Hopefully you can see that pretty clearly, that the correct answer is C, because if I have 1, 2, 3, 4, 5 on the 5th carbon, I have a hydroxy group. And so I have 5-hydroxylysine. Now keep in mind, 5-hydroxylysine, it even tells us right here, it's an unusual amino acid. It is not one of the 20 naturally occurring amino acids. Number 5 says proteins are long chains of amino acids joined together by peptide bonds. Which of the following properly depicts the peptide bond formed between 2 generic amino acids? Who could tell me which one of these is the correct answer in 5? And while you're thinking about that, I'll delete this and correct it. So the answer to 3 was C. I had written down the correct answer, but didn't highlight the correct answer. Always me. All right, so number 5, back to number 5. Could anybody tell me which one of these shows a peptide bond formed between 2 generic amino acids? Great. Ashley says B. I think B looks pretty reasonable because remember, when you're drawing a dipeptide, you'd go NCC, that represents your first amino acid, then you attach another nitrogen carbon, that's your second amino acid. The C terminus or the C terminal is a carboxylate. The N terminus or the N terminal is an ammonium. It goes NC carbonyl like this. This nitrogen should have a hydrogen. These carbons should have hydrogens, and then they both have an R group. So you see that that is manifested right here. The other ones, I'm not going to go through all of them, but you can see there's a plethora of problems in these. And so you should know how to draw a dipeptide or a tripeptide, and you should know that as long as you know that it goes nitrogen, carbon, carbon, nitrogen, carbon, carbon, and then you should be able to fill in the rest of the molecule from there. Question number 6. Tufsin is a tetrapeptide that stimulates and promotes the destruction of tumor cells. What is meant by the term tetrapeptide? Well, we've gone over this a little bit already tonight. If I have, one nitrogen, carbon, carbon, that would be the framework or the skeleton of an amino acid. If I have two linked together like this, I would call this a dipeptide. Okay, so let me just finish it off here. I'd call this a dipeptide. And so if I have 3 amino acids together, I'd call that a tripeptide. And if I have 4 amino acids linked together, I would call that a tetrapeptide. And the answer is C. It says 4 amino acids are present joined together by peptide bonds. Now, keep in mind that in a dipeptide, like I have drawn here, this is a dipeptide because there's 2 amino acids linked together here. There's only one peptide bond, right? This is the peptide bond. It's the bond between the 2 amino acids. And if you're thinking that looks like an amide bond to me, it is an amide bond, right? But when we join 2 amino acids together by an amide bond, we call it a peptide bond. Number 7 says, what is meant by quaternary structure of a protein? And I've gone over this a little bit tonight. You have to understand the difference between primary, secondary, tertiary, and quaternary structure. So again, primary structure, those are the peptide bonds that hold amino acids together. Secondary structure are alpha helices and beta pleated sheets. Tertiary structure, those are things like hydrophobic interactions, hydrogen bonding, salt bridges, disulfide bonds, polar interactions, okay, any kind of those interactions that cause the folding of a protein chain. But in order to have quaternary structure, I have to have 2 or more polypeptide chains put together to make one functioning protein unit. And that is answer E right here. It says it describes the association of 2 or more polypeptides. So what's 2 or more? 2, 3, 4, so on and so forth. Polypeptide chains to form one functioning protein unit. Question number 8 says, consider the following forms of the amino acid alanine. Which statement concerning these structures is false? Well, alanine, if I just draw the structure of alanine over here, this is probably what you get when I give you a table, for your exam, you'll have something like this. I'll give you the Zwitter ion, which means that you have the anion and the cation together. So alanine is an amino acid where the R group is a methyl. Okay, that's what alanine is. Now, all of these structures are alanine, but they represent alanine at different pHs. It says here, which one of these is false? In A, it says that structure 1, so this structure here represents the form of alanine that would be present in the blood at physiological pH. This is actually false. This is not the form of alanine that would result in or be present in the blood at physiological pH. Physiological pH is a pH of about, pH is equal to around 7.4. And what happens is, if you had this amino acid, so this uncharged amino acid, what happens is the pH 7.4 is basic enough that it will deprotonate or remove the proton from the carboxylic acid, but it's acidic enough that it will add a proton to the amino group. And so what you end up with, the actual structure at physiological pH would be what you see here, okay, what I've drawn. So this is the exact same thing as what I have drawn right here. And so that's the structure of alanine at physiological pH. And so this uncharged molecule that you see here, this would not be correct. So this is a false structure. If we look at the other answers, just to evaluate why all the rest are true, so all of the rest of these are all true. Okay, now why is that? Let's take a look at B. It says that in B, structure 2 represents the form of alanine present in the basic environment of the intestine. So if alanine is in a very basic environment, it's going to be basic enough to rip this proton off, or one of the protons off of the ammonium, and it's going to be basic enough to rip the proton or keep the proton off of the carboxyl group. And so it's going to be fully deprotonated, and it's going to be in this form here. Conversely, in C, it says the structure 3, so this structure here represents the form of alanine in the acidic environment of the stomach. Well, when it's in a very acidic environment, the carboxyl will be protonated and the ammonium will remain protonated. And so overall, it will have a charge of +1. So that's true. Structure 4 represents the Zwitter ion, and I told you what a Zwitter ion is. This is when you have a positive and a negative charge, and they balance each other out. So overall, you have no charge. Number 9 says, what is the term for the state of a protein when its organized structure has become completely disorganized? I would say that this is just a memorization question, and you should know that the answer is not hydrolyzed. Okay, it's not hydrolyzed, it's not isomerized, it's not globular. The answer is a protein becomes denatured. Say, for example, when you take an egg, the main protein that's found in egg is albumin, and when you heat that egg up, the heat actually breaks the interactions between the proteins and causes them to coagulate, and this is what we call denaturation, or we say the protein has been denatured. Also, I might have mentioned this in my slides. If you've ever, tried to grab a hot cookie sheet out of the oven and you see that your skin kind of changes color there when it gets burned really quickly by, burning it really quickly, that's denaturation. You've denatured the proteins in your skin. Anyhow, again, this is another one, number 10, that just involves some memorization. It says, what protein is the oxygen storage protein of skeletal muscles? And the answer to that is myoglobin. So myoglobin is the oxygen transport protein of muscles. Number 11, which of the following is a structural protein? I talked about structural proteins a little bit. I just told you that albumin is the protein found in eggs, so that's not really a structural protein, right? Insulin is not a structural protein as well. Neither is trypsin or myoglobin, but keratin is. In fact, if you ever listen to shampoo commercials, sometimes they might refer to keratin and say how this shampoo will strengthen keratin or something like that sometimes. Anyhow, so keratin is a structural protein. Number 12, which term best describes the relationship between L-alanine and D-alanine? Well, what do the L and the D represent? These represent the direction in which these molecules rotate plane polarized light, right? One of them is going to rotate it in one direction, and the other is going to rotate it in the opposite direction. And so the answer is that these two compounds must be mirror images of each other, right? They must be non-superimposable mirror images, rather. Then the answer to that would be enantiomers. We can write that in here. Superimposable, okay, mirror images. All right. Move on from there. This next one is kind of an interesting one. This is a tripeptide. It says, which of the statements concerning this peptide below is false? Well, you can see right away, hopefully, that this is a tripeptide. You have NCC. So here's our first peptide. Then you have NCC. Here's the second peptide. And NCC. Here's our third, I shouldn't say peptide, our third amino acid. So this is 1, 2, 3 amino acids. And so this is definitely a tripeptide, so that is true. It says its primary structure is glyprosir. I don't ask you to memorize what all of these mean. I would give you a table with these, but gly stands for glycine, and I'm sure that somebody who did the review looked this up and could corroborate that this is in fact glycine. The second one, PRO, that stands for proline. Proline is a very unique amino acid because there's no hydrogen attached to this nitrogen. It's part of a ring. It's part of this 5-membered ring, which is kind of interesting. And so this is the structure of proline. And the last one, says serine, and this, in fact, is not the structure of serine, okay? Oh, sorry, it is the structure of serine. Mr. Deon wasn't looking close enough. So this is the structure of serine. All right, so that is true, that is true. There are two peptide bonds. Yes, let's highlight them in green. The two peptide bonds, here's one peptide bond, right? It's an amide bond, and here's the second peptide bond. So it goes NC carbonyl, right? And then a peptide bond. NC carbonyl, and then a peptide bond to the next amino acid. D says the carboxyl group of the proline is joined to the amino group of the serine. That is true, right? Because we go from the proline, right? Here's the carboxyl, or the carbonyl rather, of the proline that is joined to the amino group of the serine. So that's correct. I'll delete that image for now. And then finally, it says the peptide chain contains only hydrophobic amino acid residues. That is not true, because hydrophobic means water hating or water repelling. We'll put water hating just to make it simple. And could anybody tell me which one of these is actually a hydrophilic amino acid? Which one of these would be a hydrophilic amino acid? Which one would be water loving? Thank you, Ashley. The answer is serine. All right, and I'll highlight it here. Serine is hydrophilic, water loving, right? Because serine has a hydroxyl. Serine has OH, which can hydrogen bond, and so it's gonna love water. Okay, so serine has a hydroxyl, therefore it has a polar R group, and it is hydrophilic. Number 14. What type of attractive force is responsible for maintaining secondary structure? I'm going over this many times, but primary structure are the peptide bonds. Not peptides, peptide bonds. Secondary structure is alpha helices, beta pleated sheets. And I went over tertiary and quaternary structure. The interactions that hold together the alpha helices and beta pleated sheets are hydrogen bonds between the carbonyls and the amino groups of subsequent amino acids. And in fact, there's a pretty cool figure in one of my slides where it shows a bunch of amino acids in an alpha helix like this, right? And it shows the hydrogen bonds between, say, a nitrogen and then a carbonyl down here, and you'll see the hydrogen bond like this between the nitrogen, hydrogen. Anyhow, you get the idea. So the answer is hydrogen bonds between the carbonyl and the amino groups, or the amine groups of peptide bonds. Here we go. Number 15, we have two amino acids here. The first one says ILE, that stands for isoleucine. The next one says TRP, that stands for tryptophan. And it says here, which of the following best describes where the amino acids isoleucine and tryptophan are most likely to be found in the tertiary structure of a protein? Well, when we're talking about tertiary structure, right, first we have primary structure. That's just the peptide bonds that hold amino acids together. Secondary structure, that's the result of hydrogen bonding, like we just talked about in the last question, and that produces alpha helices and beta pleated sheets. Tertiary structure, I said you can have hydrophobic interactions, hydrogen bonding, dipoles, salt bridges, disulfide bonds. But what I'm interested in here are the two R groups of these amino acids. If I draw the generic structure of an amino acid, like this, okay, and I'm sure you all know this by now, that our R group is here. And so here's the R group of tryptophan, and here's the R group of isoleucine. So if we look at these two groups, you can see that these are both, would these be considered hydrophobic or hydrophilic R groups? Who could help me out? Are these hydrophobic or hydrophilic? Not a trick question. Thank you, Tyler. These are both hydrophobic, right? Because they're just, this one's made completely out of carbon and hydrogen, and this one is mostly made out of carbon and hydrogen. Yes, you have this little nitrogen, hydrogen here, but the rest of it is all carbon atoms. And so these are hydrophobic R groups. So where are these going to be? It says at the N-terminal end of the protein. Well, not necessarily. B says at the C-terminal end of the protein. Again, not necessarily. C says on the exterior surface of the folded protein where the contact with water is the greatest due to their hydrophilic nature. Well, that's not going to be true, right? We just said they're both hydrophobic. They're water-hating. What about D? In D, it says in the interior of that folded protein away from water due to their hydrophobic nature. Exactly, that would be the answer. And so when we have tertiary structure in a protein, if you have a big protein chain and it's all folded up into some kind of tertiary structure like this, forgive me, that's my ribbon diagram. Maybe I should have put an alpha helix in there. Anyhow, the water molecules are going to be on the exterior. Now, what I've drawn here is just random, but you know that tertiary structure is not random, right? Proteins have evolved so that they fold in such a way to produce the desired effect of that protein. So all of the amino acid groups that are water-loving are going to be on the exterior of the protein, and all of the water-hating or hydrophobic amino acid residues, those are all going to be found in the interior. And so the answer to 15 will be D. Since both of these R groups are hydrophobic, they're going to be on the interior of the protein, away from the water. The next question says, or asks us to draw the structure of phenylalanine, draw the form of the amino acid that is expected to predominate at the stated pH. All right, I'm going to change this one a little bit because I looked at it this evening and I thought it was a little bit tricky the way it's written. So I'm going to make it even simpler. And if you're like, hey, I already did the question, hold the phone before you make any harsh decisions, okay? What we're going to do is we're going to do something a little different, okay? We're going to cross this out. We're going to draw, and we can do it as a group, okay? We're going to draw phenylalanine, okay? And we're going to draw A. We're going to draw it at pH is equal to 1, and then B we're going to draw it at pH equal to 11. Okay, so we're going to make it really simple. So this is even simpler than the questions I had here. I think these ones are too complicated. So first I'm going to help you out by drawing the structure of phenylalanine. So we have our amino group, we have our carbon, hydrogen, and we have our carboxylate. Phenylalanine is when you have a CH2 and then you have an aromatic ring like this, okay? So if I want to draw the structure of phenylalanine at a pH equal to 1, a pH equal to 1 is very, very acidic, right? This is strongly acidic. Strongly acidic. And so what does that mean? In an acid, if it's in an acidic environment, protons will be donated to the molecule. Where can this molecule accept protons? I cannot accept any more protons at this end because I already have a positive charge. What about on the C-terminus? Can that end accept a proton? Who could help me out? Yes or no? Could that end accept a proton? Can the carboxylate accept a proton? Yes, absolutely, Tyler. Thank you. It can take on a proton. And so at a very acidic pH, the structure of phenylalanine will look like this. It's going to look like this. So overall, it's going to have a charge of +1. If I put it in a strongly basic environment, right, like a pH equal to 11 or 12 or 13, we'll just put here strongly basic, or strong base, okay? What's going to happen then? Could anybody describe it? I know it might be a lot to type out, but can anybody just try and describe what's going to happen? I'm going to put it in a very basic. I love the rip off the proton from NH3. You're absolutely right. Right? We can't remove any more protons here. There's none to remove. But we can remove a proton off of there, right? So let's do that. So what we're going to end up with is, instead of NH3+, we're going to have NH2 bound to the carbon. We're still going to have our carboxylate like this. We have our methylene, and then we have our aromatic ring. And so overall, we're going to have a charge of -1. So I think that actually lowers the complexity a little bit than the questions that I had asked. And that's kind of the complexity that I would expect you to, have down pat for the upcoming exam. Let's take a look at the next question. It's kind of a fun one. It says, what does it say? It probably says all kinds of stuff. It says, using 3- and 1-letter abbreviations, show the sequence of amino acid residues in the following pentapeptide. So if it's a pentapeptide, then you know that there are, penta means 5. There are 5 amino acids here. Again, you don't have to memorize the structures of the amino acids. I would give you a table to help you out on this. So it goes NCC. So here's my first amino acid. Then I have NCC, second, third, fourth, fifth. I bet you somebody in the class tried to identify all these, and I hope you all looked at the book to get some help with that. Because in my slides or in my videos, I told you don't have to memorize all these. Could anybody help me out with the first amino acid? Tell me what the first one is. Not a trick question. Yes, absolutely. The first one is leucine, and the 3-letter abbreviation for that is LEU. So we'll put here leucine. What about the second one? Yes, the second one is alanine. Alanine. Okay, and if you're wondering, how did Ashley know this? Well, again, if we have our amino acid, a generic amino acid, just an amino acid you see walking down the street, okay? We draw it as a zwitterion like that. What makes amino acids unique are their R groups. When R is equal to a isobutyl group, so when R is equal to an isobutyl group, like is shown here, then it's leucine, right? So when R is equal to isobutyl, we have leucine. When R is equal to a methyl group, you have alanine. Again, you don't have to memorize these, but that's how she's figuring this out, right? Because I have a methyl group here. Now I have another question for you. What if my R group is a CH2 and an aromatic ring? Who knows what that one is? Again, exactly. Thank you, Tyler. Yes, it's phenylalanine. Absolutely. So this is phenylalanine. The abbreviation we use is PHE. So that's phenylalanine. Anyhow, I'll help us out with the rest of them just to kind of get through it. The next one is cysteine. So CYS. Should pronounce it cysteine. And the last one is aspartic acid. So ASP is aspartic acid. Anyhow, so you just have to be able to identify these amino acids by looking at their R groups. And I'll circle all of the R groups if you want to go back and look at them again. Number 18. It says, determine which of the following peptides will have a higher molecular weight. I don't think I would ever ask this question in 100 years to any of you. I just kind of put it in here as an interesting, just sort of a fun thing. You just have to compare the residues of the amino acid, or compare the residues of the R group. Anyhow, so maybe we'll skip that one for now. I might look at that one tonight. Yeah. Also, yeah, we've already gone over 19 or something like it that I would ask. Don't know what kind of phenylalanine. So let's take a look at number 20. Yeah, number 20. I think number 20 is a good question. It says, just get some practice in drawing a pentapeptide. So it says methionine enkephalin is a pentapeptide that is produced in the body to control pain. From the sequence of its amino acid residues, draw a bond line structure of methionine enkephalin. Okay, if I've got follow me very carefully here. I've got 5 amino acids, right? It says here pentapeptide. So I'm going to start like this. Nitrogen, carbon, carbon. There's my first amino acid. Nitrogen, carbon, carbon. There's my second. Nitrogen, carbon, carbon. There's my third. Carbon, carbon, and nitrogen, carbon, carbon. Okay? There's my skeleton for my 5 amino acids. On the C terminal, or the C terminus, I'm going to put my carboxylate. And on the N terminus, I'm going to put my ammonium. Next step is to go NC carbonyl, NC carbonyl, NC carbonyl, NC carbonyl. And the last carbonyl, of course, is part of a carboxyl. Okay, I've got all that done. Next thing is to put a hydrogen attached to each one of these nitrogens, like this. I'm also going to put a hydrogen attached to each one of the carbons that's left over. And the last thing I have left to do is to fill in the R group. So 1, 2, 3, 4, 5 R groups. Okay, first we'll do a tyrosine. A tyrosine, again, you'd have to look this up because I don't expect you to memorize all of the amino acids. Here's a tyrosine. The next one is a glycine, and then one following that is a glycine. Luckily, glycine is the easiest amino acid in the world because the R group is just a hydrogen. So we'll fill out both of those. The next one is a phenylalanine. And in a phenylalanine, where am I? We have a CH2 and our phenyl, like this, or our benzene ring. And the next one is MET, which stands for methionine, which has 2 CH2s, sulfur, so it's a sulfide, like that. Okay, so this is the R group of tyrosine. Hydrogen is the R group of glycine. The CH2 followed by an aromatic ring is the R group of phenylalanine. And this sulfide is the R group of methionine. Again, if you're a little confused, I'm going to point out here's our first amino acid, here's our second amino acid, here's our third, here's our fourth, and here's our fifth. Could anybody tell me how many peptide bonds are in this pentapeptide? How many peptide bonds? That's right. Thank you, Kamish. Thank you, Tyler. Exactly. There are 4 of them. You're unsure. Let's highlight them. Okay, I have 1 here, 1 here, 1 here, and 1 here. So that's a total of 4 peptide bonds. Those are the amide bonds that link these amino acids together. Let's move on to chapter 19 and try some more practice. Question number 1. The pancreatic enzymes trypsin, chymotrypsin, and elastase all hydrolyze peptide bonds on the carbonyl side of several different amino acids in a protein. Which term, or what term best describes their enzyme specificity? I'm going to leave this up to you guys. Does anybody have the answer to this one? And I'm just going to do some of my artistry in the meantime. Can anybody tell me what type of specificity it has? If it says that what's being hydrolyzed, right? Hydrolyzed means being broken. If you're hydrolyzing peptide bonds, that's the key word, is bonds, okay? So it's not stereospecific because we're not even talking about the chirality. They're not peptide specific. They're not absolutely specific. What are they specific to? They're specific to the linkage, because linkage is described a type of bond. And so the answer is linkage. And we could even write that in here. So type of bond. Okay. All right. Question number 2 says, what is an allosteric enzyme? And I went over in my videos all kinds of information about enzymes, and then I spoke about allosteric enzymes, and I spoke about positive effectors and negative effector molecules. And basically, what an allosteric enzyme is an enzyme that has an active site where you'll have your substrate bind, and then it becomes an enzyme substrate complex, and then it, you lose the product. Okay, but the difference is between a regular enzyme and an allosteric enzyme is that an allosteric enzyme has a second binding pocket, or a second part of the molecule, where a small molecule can bind to either turn the enzyme on or off. And so the answer to number 2 for chapter 19 is an enzyme whose activity is actually regulated by the binding of a small effector molecule. Number 3 says, which of the following correctly describes pepsinogen? And you probably remember, if you watched my videos, I talk about inogens a little bit. And whenever you see inogen, okay, if you ever see the suffix inogen, it means an inactive form of an enzyme. Inactive form of enzyme. And so if you hear of pepsinogen, chymotrypsinogen, so on and so forth, pepsinogen is the inactive form of the enzyme pepsin. Okay, so you have to know what the inogens are. Number 4, what 3 structurally similar pancreatic serine proteases function to cleave certain peptide bonds in proteins? I'm going to see who is watching the videos and who could answer number 4. Who could help me out with number 4? Somebody says D. I think you are correct. Okay, the pancreatic serine proteases are trypsin, chymotrypsin, and elastase. Number 5 says trypsin is a protease enzyme that cleaves peptide bonds on the carbonyl side of basic amino acids, and that's going to be important. So I'm going to underline it right away. Basic amino acids. Which labeled peptide bonds in the peptide below would be cleaved by trypsin? So basically, if you were to take this peptide here and you threw it in a reactor and you added some trypsin to it, who could tell me which one of these bonds is going to be broken? Would it be A, B, C, or D? Which one of those bonds would get cleaved by the enzyme trypsin? Sorry, I'm going to open my chat here. Somebody says C. Okay, so let's take a look here. Make sure it's correct. Okay, so first let's identify the peptide bonds. Okay, A is a peptide bond, B is not a peptide bond, C is a peptide bond, and D is not a peptide bond. So then it boils down to, you can say A and C are peptide bonds. Okay, so here B and D are not peptide bonds. Okay. Well, it says here it's on the carbonyl side of basic amino acids. Okay, if I look at this residue, this is acidic, right? This is a carboxylate, which is the conjugate base of an acid. So this comes from an acidic residue, whereas this comes from, or this is a basic residue. Okay? In fact, the second amino acid here, I'll help you out. This is lysine, which is a basic amino acid. Anyhow, so if it goes NCC, okay, it says it breaks it on the carbonyl side of the basic. So this is the N-terminus side, this is the carbonyl side. And so the answer must be C. It breaks this bond here, C. Oops. Didn't mean to do that. I'll clean up some of this spinach here so it's a little bit tidier. Anyhow, so the bond that we end up breaking is C. Number 6, lactase is an enzyme that catalyzes the following reaction. Which statement describing this reaction is false? So we have lactose. We have lactose, which is shown here. It gets acted upon by the enzyme lactase, and we produce glucose and galactose. Those are the two monosaccharides we get when the disaccharide lactose undergoes hydrolysis by the enzyme lactase. Let's take a look at the answers and see if we can figure out which one of these is false. Remember, we're looking for the false answer. It says lactose is the substrate. That is true. This is our substrate, which would bind to the active site in lactase, our enzyme. It says B, glucose is a product. Well, obviously that's true. You can see glucose is right there. D, galactose is a cofactor. Galactose is not a cofactor in this reaction. Okay, the reaction occurs at the active site of lactase, of course, and lactase is a hydrolase enzyme. Well, that's true because we end up breaking the glycosidic bond here in lactose. So galactose is not a cofactor. A cofactor is a small molecule or ion that is required in order to activate an enzyme. Number 7, what is the term for a molecule that is similar in structure and charge distribution to the substrate in an enzyme catalyzed reaction? So if we have a molecule that is similar in structure and charge distribution to the substrate in an enzyme catalyzed reaction, I remember I had this in my slides, and this is the kind of way that the book had it. So say you have an enzyme, and let's say you have, a binding pocket that looks something like this. And let's say the actual substrate kind of looks like this. Okay, so you can see how the substrate, the topography of the substrate here fits the binding pocket. Okay, so there's my substrate. But if you have something that's similar in structure and in charge distribution, okay, so if I have a molecule that looks kind of like this, okay, it's got like it would fit the binding pocket. Maybe I can make that better. Sorry, Mr. Deon is not a very good artist, you guys. Okay, but then it has something like this, but it still has that portion that is going to fit into the binding pocket. Okay, who could tell me what we call this kind of molecule? It's not identical, but it does have a part which is almost identical, right, to the actual substrate molecule. Could anybody help me out with that? Thank you, Tyler. It's a structural analog. That's exactly right. And a good example of a structural analog would be a drug. Okay, a drug is a very common example of a structural analog, right? We model many drugs, or we try to get part of the structure of the drug to mimic the structure of an actual substrate that can bind to an enzyme. And we might want to elicit the response of that enzyme, or we might want to inhibit the reaction that enzyme is responsible for. Thanks. Number 8. What class of enzymes catalyzes the following reaction? Well, you don't really have to know a ton about enzymes to see that this molecule right here, this is a TGC. Right? This is a triglyceride. Oops. Okay. This is a triglyceride. And I break it apart to give me glycerol. Everybody in the class should know the structure of glycerol by now. And a fatty acid. Fatty acid, okay? So what kind of enzyme is catalyzing this reaction here? Well, we've got hydrogenase, catalase, isomerase, elastase, and lipase. Who could help me out? Which one do you think is the best answer here? Which one do you think would be the best answer? Absolutely, right? Because you think of the word lipid, right? And A triglyceride is a lipid. So a TGC is a lipid. And so we're breaking apart a lipid. And so this is a lipase. And you can see which bonds very clearly that we're breaking these carbon-oxygen single bonds to produce glycerol and the fatty acid. So a lipase acts on a lipid, in this case, a triglyceride, and we end up with glycerol and 3 fatty acids. Number 9, elastase is a protease enzyme that cleaves peptide bonds on the carbonyl side of the amino acids glycine and alanine. So on the amino acids, on the carbonyl side of the amino acids glycine and alanine, what products result from the treatment of the peptide, with the primary structure shown below with elastase? So what this question is asking, if you're like, I'm not sure what's going on here, what it's saying is, let's say you have this tetrapeptide, okay? You have it in a round bottom flask. I'll even do you one better. I'll draw a round bottom flask. Okay, here you are, you're in the lab, and you have that phenylalanine, alanine. Sorry, my flask isn't big enough to hold it, is it? Anyhow, what would happen if you took elastase, and you just threw it in there? You just threw it in there, and then you stirred it around. What are you going to end up with? Well, it's giving you a clue here, okay? It says protease. What did I say? Elastase? Oh yeah, sure. Elastase. It says elastase cleaves peptide bonds on the carbonyl side of the amino acids glycine and alanine. Okay, we don't even need to draw the structure of this tetrapeptide. I want to ask you guys, was anybody able to figure this one out? Could anybody tell me what the answer would be for this one, number 9? I'm just curious to know if anybody got the answer without drawing it. I was thinking D, but I'm not sure. I was thinking D, but I'm not sure. Okay. I think that looks pretty reasonable, right? Because if you have proline, glycine, phenylalanine, alanine, remember, let's draw it out, okay? We got time. I'm going to draw the tetrapeptide. I'm going to have to kind of squeeze it in here. NC, carbonyl, NC, carbonyl. I got one more to go. NC, carbonyl. Now, again, you don't have to do this. You don't have to draw the whole damn thing out, but I'm going to do it because I'm the instructor. Okay, I'm going to fill in these hydrogens and the nitrogens. And so proline, I already got a problem here because I have to fix up my proline. Anyhow, no problem. Because proline is part of the nitrogen is part of a ring. So it looks like this, like that. So instead of having there, it looks like this. So this is my proline. The next one is phenylalanine. They're all big. Anyhow, I'll put aromatic ring down here. The next one is, oh shit. The next one, sorry, pardon my language. The next one is glycine. I think Mr. Deon needs some supper. All right, or what do you guys call it? Dinner down here? Yeah, food. And the last one is alanine. Okay, I promise. I swear it's good now. All right, it says on the carbonyl side of glycine and alanine. So here I have proline, so forget that, okay? The next one is glycine. Where's the carbonyl and glycine? Here's my carbonyl and glycine, so it means it's going to break the bond there. Then I have phenylalanine, and I have alanine. Well, alanine, the carbonyl of alanine is right here. There's no bond to break. So what are you going to be left over with? You're going to be left over with proline attached to glycine, and phenylalanine, alanine. And so you guys, whoever answered me, was totally right. You end up with proline attached to glycine and phenylalanine attached to alanine, like that. Give me a thumbs up if you're like, okay, I get it now. It makes some kind of sense. While you're doing that, I'll take a drink of bubble water. Okay, good. All right, fantastic. Great. Again, if you have any questions about that, feel free to ask me tonight in the lab. I'd love to talk to you about anything to do with amino acids. Number 10 says, which of the following statements concerning enzymes is false? It says enzymes are the body's biological catalysts, increasing the rate of cellular reactions. Well, if I had to come up with a definition of enzymes, I'd say that's a pretty dang good one. So that is certainly true. Enzymes are usually globular proteins with flexible three-dimensional shapes. Of course, most enzymes have primary, secondary, and tertiary structure, and the tertiary structure is that globular structure, and we know that there's some flexibility once they adopt that three-dimensional shape. That is true. Letter C says the reactant in an enzyme catalyzed reaction is called a cofactor. That's not true. The reactant is actually called a substrate. And so the answer to this question must be C. Let's look at the other ones just for fun. D says the active site of an enzyme is where the substrate binding and catalysis occur. That is definitely true. And the last one, E says inhibitors are molecules that cause an enzyme to lose its activity. That's exactly true. Number 11. Proteases or proteolytic enzymes are responsible for which of the following functions? Who could help me out with this one? I think this one is pretty doable. Which one of these is the best description of what a protease is? Ashley says C. That's exactly right. A protease will cleaves peptide bonds in proteins. There we go. Boom. Hydrolysis of peptide bonds between amino acids in proteins. Thanks, Kamish. Thank you, everybody. I realize, one thing I've learned about teaching on Teams is that some people, all of our internets aren't equal speed. Sometimes I'll ask a question and wait, a few minutes to get a few seconds to get an answer. Okay, let's move on to chapter 21. Chapter 21. Okay. I guess I kind of have these questions numbered a little funny, but let's go to question number 12. It says, in what are the two ways in which the energy of a food source can be released in a catabolic pathway? Who could answer this one for me? We know that an anabolic pathway, that's building up, and a catabolic or catabolism is breaking down. So how can we release the energy of food source in a catabolic pathway? Who could help me out? I think Tyler answered the question, and the answer is C. Okay, the answer is C, which is chemical bond energy and heat. You think about the human body, it's nothing more than a big combustion chamber. And what it means by chemical bond energy, if you're thinking, does that mean ATP? Yes. It's adenosine triphosphate and heat, okay? So when we consume food, we store some of that chemical energy as the bonds in ATP, the high energy phosphoanhydride bonds in ATP, and then we also convert a lot of it into heat to keep ourselves warm. Number 13 says the abbreviated form of the equation for the first reaction of glycolysis is shown below. What is the missing product in this reaction? And so I think it's a good idea to have the first reaction of glycolysis memorized. I think you should memorize what happens in the first step of glycolysis, the first reaction. And what's happening here is that we have glucose plus an inorganic phosphate plus some energy, produces water. And who watched the videos or read the book enough to know what molecule we end up producing in the first step besides water? Somebody said, Tyler says B. Okay, the answer is glucose 6-phosphate, and that is correct. So we end up producing water in glucose 6-phosphate, which we abbreviate G6P, glucose 6-phosphate. Number 14, which pathway is not correctly identified as catabolic or anabolic? Remember, anabolic means building up, building, and catabolic We could describe that as decomposing or breaking down. We'll just put breaking down. Breaking down. Okay. So digestion of starch is catabolism, or is a catabolic process. That is true, right? Because digestion is breaking down. B says the synthesis, so the building of proteins from amino acids, is anabolic. That is also true. C says the oxidation of glucose to CO2 and water is a catabolic process, right? That's basically burning it, right? We basically burn glucose in our bodies. So that is catabolism. That is a catabolic process. The formation of ADP from ATP is an anabolic process. Well, if we think about it, when we take ATP and we break the phosphoanhydride bond, or one of the phosphoanhydride bonds in ATP to produce ADP, we release energy, right? Because we're breaking down the ATP. So that is actually a catabolic process. And so this is false, and that would be the correct answer because we're looking for something that is not correctly identified. And let's just take a look at the last one. It says the conversion of glycogen to glucose is catabolism, or is a catabolic process. That's absolutely true, right? Glycogen is actually glucose stored in our body. It's one of the ways that we store glucose in our body. And so we're going to say T, that is true. Number 15 says, what is the first reaction in glycolysis which traps glucose within the cell? This one I'd say is nothing more than a memorization, of what happens, right? We said we take the glucose, let me back up here. We take the glucose, we treat it with the inorganic phosphate, and we make G6P. So basically, when you add the phosphate group, we call that phosphorylation. And when you add that phosphate group, that's what traps it inside the cell. You might have had to pay close attention in the videos or in the textbook, in your reading, to get the answer to that one. But the answer is the phosphorylation of glucose is what traps glucose within the cell. Number 16 says the hormones glucagon and epinephrine primarily control which biochemical pathway? Who was able to figure this one out? I think somebody put C. Sorry, I'm rifling back and forth here. Anyhow, it's glycogenolysis. That's another one you kind of have to memorize. Let's move on from there. What do we have left? Oh, we only have a few. Let's see. These last ones, they have a lot of reading to get through these. Anyhow, number 17 says the hydrolysis of ATP, or adenosine triphosphate, can be used to drive a reaction. What does that mean? It says the ATP acts as a carrier molecule for substrates. That's got nothing to do with driving a reaction. B, the ATP produced upon hydrolysis acts as a catalyst to increase the rates of otherwise slow biochemical processes. Well, that's not really about driving a reaction. That's more about catalyzing a reaction. C says the energy produced from the hydrolysis of ATP is used by energy requiring reactions of the body. I'd say that looks like the correct answer to me. Because when we hydrolyze ATP, we produce energy, and we use that energy to do all kinds of different reactions in the body. And in fact, one of the first things that I talk about in the videos for chapter 21 are all the bodily functions that require ATP to occur. Number 18, which of the statements concerning gluconeogenesis it says, which of the statements concerning gluconeogenesis is false? Gluconeogenesis, what is gluconeogenesis? We think of glucose, new, genesis, making. Right, so gluconeogenesis is making glucose from non-carbohydrate or non-carbohydrate starting materials. I'm getting tired here. Anyhow, so the definition of gluconeogenesis is actually A. This is true. Gluconeogenesis is the process by which glucose is synthesized from carbon atoms from non-carbohydrate sources, okay? Gluconeogenesis B is the pathway that protects the brain and blood red blood cells from experiencing a loss of glucose. This is also true. It says here in C that gluconeogenesis is the reverse. The gluconeogenesis pathway is the reverse of glycolysis. That is false. It is not true. And in fact, I go over this in my videos, how steps 1, 3, and 10 are catalyzed by different enzymes in gluconeogenesis. So if you just think about gluconeogenesis as being the reverse of glycolysis, some of the enzymes in the two pathways are similar, but steps 1, 3, and 10 use different enzymes. And so gluconeogenesis is not exactly the reverse pathway of glycolysis. Let's move on to question number 19. Step 3 in glycolysis is shown here. We have F6P, which is fructose 6-phosphate. It takes an inorganic phosphate from ATP, transfers it to make F16BP, so fructose 1,6-bisphosphate. Now we have 2 phosphates, and in the process, we make good old ADP. Okay. I'm getting tired from reading. Who could tell me which one of these is the true statement? A, B, C, D, or E? Ashley says A. Let's take a look. It says the hydrolysis of ATP. Yes, because ATP is being hydrolyzed to form ADP. The hydrolysis of ATP provides the phosphoryl group. That is correct, because we go from adenosine triphosphate to adenosine diphosphate. So we did remove a phosphoryl group and the energy necessary for the formation of F16-bisphosphate. That is correct. Absolutely correct. Okay. When we break that ATP, we end up getting energy. We talked about that earlier tonight. And we end up with the phosphoryl group. And both of those, the phosphoryl group and the energy, are what is used to put the phosphoryl group onto this hydroxyl here to make a bisphosphate. Perfect. Let's look at the last two, and then we'll call it quits. And then we'll meet up at 8 p.m. at Rampart Range. Number 20. Let's go over these. Which statement concerning glycolysis is false? Glycolysis is an anaerobic process that occurs in the cytoplasm. That is absolutely true. Glycolysis is an ancient mechanism for the metabolism of, or the breaking down of glucose. Glycolysis requires energy provided by ATP. That is absolutely true. I talked about how you have the investment phase in glycolysis, and then you get the payout in the long run. In glycolysis, it says glycolysis in C is a pathway that consists of 10 enzyme catalyzed reactions. Yes, I referred to the 10 steps earlier. Glycolysis is the final step in the process to produce energy from food. That is absolutely false. That is not true. Okay. Let's see. Number 21. We just moved down here. Oops. I got a problem with slides. All right, let's take a look at the last one. It says, in what way is the reaction shown a key one in carbohydrate metabolism? I have pyruvate. I'm treating it with acetyl coenzyme. Acetyl coenzyme A. My NAD plus is being reduced to NADH. My enzyme is pyruvate dehydrogenase. Sorry. I'm taking my pyruvate, I'm adding it to coenzyme A, and I produce acetyl coenzyme A. Anyhow, I think I'm too tired. All right. Who could tell me which one is the correct answer here? Who could help me out with 21? Well, if you're unsure, I can help you out. The answer is A, because we need to get pyruvate into the citric acid cycle because it's one of the products of glycolysis. And this is how we get our pyruvate into the citric acid cycle, where it can be completely oxidized to produce 1-2 molecules of carbon dioxide in the end. Anyhow, that covers all the questions. So what I would like, let me just stop